Welcome to our class Neet Biology, and today let us discuss DNA fingerprinting, widely used in forensic science. Now, what do you mean by this DNA fingerprinting means? It is the uniqueness of DNA which is present in the individual, and this is widely used in forensic science. This technique was first proposed by the scientist Alec Jeffrey at the University Leicester in 1985. So this technique DNA fingerprinting is also called as DNA profiling or molecular fingerprinting. Now, what is the principle present behind this DNA fingerprinting means? The variable number tandem repeats. Okay, now all the information of DNA is present in the form of nucleotides, that is adenine, guanine, cytosine and thymine. So all the entire DNA is divided into two regions. They are the coding regions and non-coding regions. Coding region consists of the genes and they are responsible for synthesis of proteins. Whereas the non-coding genes are also called as the junk DNA, which are not responsible for any protein synthesis. And now the current research on junk DNA is made clear that it helps in maintaining the structure and function of the DNA. Okay, and also it sheds the light upon the evolutionary process. Okay, now these repetitive sequences which are present in the junk DNA is not same in any two individual. It is unique to each and everyone unless until they are homozygous twins. These homozygous twins only have that same set of tandem repeats or repetitive sequence. Other than that, no two individuals will have the same tandem repeats in the DNA. So this tandem repeats from where the child acquired means 50% of this repetitive sequence in the DNA is being inherited from the mother, and other 50% of this repetitive sequence in a particular chromosomes are being inherited from the father. Now, how the DNA is being extracted? It means how the strategy of DNA fingerprinting is being done means the protocol for that is firstly there should be extraction of DNA. Okay, then after that the DNA should be treated with restriction endonucleases. The DNA should be separated by gel electrophoresis. After that, the bands of DNA should be transformed onto the nitrocellulose membrane, and these bands are being illuminated under autoradiography. Now the DNA extraction method. Okay, the cells are broken up and the DNA which is present in the nucleus is being isolated. And this sample, either it may be the blood, hair, saliva, semen or any body tissue which we get from the crime scene can be collected as a sample. And after that, the DNA which is isolated should be treated with a restriction endonucleases. These restriction endonucleases are also called as molecular scissors, which are responsible for breaking down of DNA at a particular target site. So the fragments of DNA are being attained by using this restriction endonucleases. After DNA cutting treatment with endonucleases, that samples are being separated by using gel electrophoresis. After separation, we can get the bands or the tandem repeats like this. And here the DNA separation occurs depending upon the size of the DNA. Okay, by this we can say, okay, that you know that the DNA is having the negative charge. So when the electricity is being applied, charge is applied, automatically this move towards the positive charge. Okay, the shorter fragments will be moving fastly than that of the larger molecule. So this is the separation technique which is depending upon the molecular weight. Next, after separation of these DNA bands, they should be transferred onto the nitrocellulose paper or the nylon membrane. So this is the gel onto which the bands are being separated. And this gel is now placed by the nitrocellulose membrane and filter papers. So that whatever the DNA content present in the gel, by diffusion it reaches this nitrocellulose membrane or nylon membrane. That separation of DNA is called as Sutherland blotting, as that was being invented by the scientist Sutherland. So here on to this nitrocellulose paper or nylon membrane, here you can see how the DNA fragments are being attached. So here this is the DNA which is being transferred onto the nylon membrane. And after this, the bands which we get on the nylon membrane are being placed beneath the X-ray film. And when light is passed, that is the radiography is being done, we can see the bands which are being illuminated under this auto radiography. Okay, now the patterns of these bands will be compared with the suspect in order to know who the person exactly has committed the crime. Here, let us take an example. Let us think that there is a violent murder which has been occurred, and this forensic team has been retrieved a blood sample from that crime scene. And they have prepared the DNA profiles of blood sample, and the victim and the pictures has been given like this. Okay, now here this is the victim profile and this is the blood sample which they have collected from the crime scene. So these are the repeats, tandem repeats of a particular chromosome which they have been assayed. And this pattern should be compared with the suspect. So now if you see there is 100% matching with that of the suspect, then this is the evidence for the person to be sentenced out. And this is most beneficial for using this in paternity suits as well as inheritance cases and immigration cases. Now, if suppose let us assume that the man is claiming that this child does not belong to him. Now, if we do this DNA fingerprinting, the child DNA should match with the mother as well as the father, because he inherit 50% from the mother and other 50% from the father. But he is not exactly the Xerox copy of either the mother or father. 50% is from the mother and the other 50% is from the father. Now, if you check the tandem repeats which are present here or the bands which are being present here, you can see. Okay, this is the perfect match here with the mother, and again at this place there is matching with the mother. Okay, so now come towards the other side, towards the father. So here there is again matching with the father. Okay, so here also 50% there is matching with the father. So we can say this man is the biological father to this child. The famous cases which are being solved by using DNA fingerprinting after its invention is, that is the Elizabeth Hurley, who used this DNA fingerprinting in the year 2002 to prove that Steve Bing was the father to her child. And later on in the year 1986, there is one more case which was being held by using this DNA fingerprinting, and that is pitchfork. He was the first criminal who was caught based on this DNA fingerprinting as an evidence. He was arrested in the year 1986 for the rape and murder of two girls and was sentenced in the year 1988. If you like the video, share and subscribe. Press the bell icon to receive further notifications. This is Pravalika signing off.